Good evening and welcome to this program, Microbes in Seeds. I'm Laurel Westendorf. I'm part of the Community Relations team at the Deschutes Public Library. Every month we explore a theme in our programming, and this month's theme is mystery. For more free library programming, go to our event calendar found at deschuteslibrary.org/calendar or find recordings of programs on our library YouTube channel. Our presenter is Jillian Bergman. She's a second-year ecology PhD student at the University of California, Davis. Jillian is a recipient of the National Science Foundation Graduate Research Fellowship and the UC Davis Dean's Distinguished Graduate Fellowship. She received her honors Bachelor of Science in Bioresource Research at Oregon State University. Thank you so much, Jillian, for presenting tonight on microbes in seeds.
Thank you for having me. It's exciting to be here. Let me just share my screen. Can all of y'all see that? Perfect. Hi, everybody. As Laurel said, my name's Jillian. I'm a second-year PhD student in the lab of Johann Laveau at UC Davis. And tonight, I'm very excited to talk to you about the mysteries of seed microbes. And so before we dive in, I wanted to give a quick outline of what I'm going to talk about tonight. First, I'm going to start with why seeds and their microbes are important. Then we're going to look at what seed microbial communities look like, how microbes get into seeds in the first place, and what factors are important during that process. And finally, what do microbes do once they're inside seeds? And then some concluding thoughts. And so to start, why are seeds and their microbes important? And I think it's important to start with what we think of when we hear the word seed. And I think for a lot of people, when they hear about seeds, they think of food, right? So crops like corn, soybean, rice, wheat, other grains and legumes. These are all examples of seeds that we eat every day. And seed crops are part of what allowed humans to transition to the societies that we have today through the development of agriculture. And so our relationship with seeds is thousands of years old and fundamental to our social evolution. And the majority of the calories we eat in a day come from seeds or some seed-related product. But what are seeds in terms of plant biology? So seeds are the agents of plant sexual reproduction. Seeds are structures that we see in flowering plants like this almond plant here with this friendly little bee. You also see seeds in gymnosperms, such as conifers like this pine here. And seeds have three main parts. They have a seed coat, which protects the inside of the seed. There's an endosperm, which is a compound of nutrients that nourish the young plant. And then finally, we have the embryo on the inside, which is the future plant that's going to germinate and grow out. And seeds have a variety of ecological, economic, and cultural values. Because they're a product of plant sexual reproduction, seeds are really important in the maintenance and regeneration of plant populations. And because many of them are small, seeds allow plant populations to disperse or move around in space and colonize new areas, which is really important for plant ecology. And because mature seeds are able to sit dormant in the soil, many of them can withstand greater environmental stressors like drought or temperature extremes than their adult counterparts can. And some seeds even need extreme conditions to be dispersed and to germinate, such as lodgepole pine, which is shown here, whose cones can only open after wildfire events. And because seeds are produced through sexual plant reproduction, they're also very genetically diverse, or they have a diversity of traits. And this diversity is essential to plant survival against disease, nutrient limitations, competition with other plants, and environmental stressors. And this diversity is also important for crop plants in what's called agricultural biodiversity. And Dr. Nabhan defined agrobiodiversity as the cornerstone for building greater food security for humankind. And this is why we have seed banks built all over the world to maintain a variety of seeds, plant species and seeds of different varieties within a plant species, so that we can protect that wild and crop biodiversity in the face of threats such as climate change. And in the face of climate change, we have to think about all the factors that contribute to plant survival and health, including that of seeds. And one really important factor is the relationships between plants and their microbes, such as fungi, bacteria, archaea, and other microorganisms. And the most well-known example of plant-microbe relationships are mycorrhizae, where plant roots join with fungal filaments, also known as hyphae, to exchange water, minerals, and sugars. And about 90% of land plants have relationships with mycorrhizal fungi, including forest trees like this one drawn here, and various crop plants. And this relationship has been around for millions of years. So this figure is showing fossils of 400 million year old early land plants. And the little arrows are pointing out the fungal hyphae growing in the above ground and the below ground tissues of these plants. And this demonstrates that mycorrhizal fungi were essential in the establishment of plants on land. And so this relationship has contributed to the great plant diversity that we've seen today, which in turn impacts animal and human life. However, there are many other relationships between plants and microbes that span all plant tissues, and they impact many plant functions. For example, bacteria and yeast fungi are found on flowers and in flower nectar, such as these bacteria here growing on a flower petal. And they can impact pollinator attraction to flowers and pollen germination. Bacteria and fungi are also found in and on leaves, where they can modify disease development, such as this mold that's parasitizing a rust pathogen or disease-causing agent. And they can also modify nutrient uptake. Finally, bacteria, fungi, and other microbes are found in, on, and around roots, where they can enhance nutrient uptake, alter disease development, and improve tolerance to stressors like drought. And as with all these other tissues, seeds host microbes, and the role of these microbes in plant health is of growing interest for several reasons. The first main reason is that seed microbes can be vertically transmitted. And what I mean by that is you'll have a variety of microbes living in a mother plant, and when that mother plant produces seed, some of those microbes are transferred from the mother plant into the seed. And when that seed germinates, those microbes are transferred from the seed into the developing seedling. And so what we have is a multi-generational relationship between plants and their microbes. Seed microbes are also really interesting because seeds have a heightened mortality risk in the face of pathogens, pests, and abiotic stressors. Therefore, interactions with microbes can either significantly improve or hinder seed and seedling survival. And a lot of these interactions include those with putative pathogens, like this Pseudomonas bacterium that's growing out of and killing these poplar seedlings. And these interactions with pathogens can maintain plant diversity via infecting susceptible seeds while not infecting resistant seeds. However, seeds also have interactions with neutral or beneficial microbes, such as the microbes that were here inoculated on these seedlings, producing better growth. And I first became interested in seed microbes as a college student at Oregon State University. So I got my undergraduate degree there in the agricultural sciences, where I was broadly interested in the intimate relationships between fungi and plants. And for my degree, I had to do a thesis research project, and I chose to do my project with Dr. Posey Busby, who's here next to me. We're looking at a poplar leaf. And I began working with her to look at the seed fungi inside poplar trees. And then for my thesis project, I surveyed the seed fungi inside Douglas fir, such as the seeds in this cone here. And in my thesis project, we sampled seeds from sites across the Pacific Northwest, as well as a site in New Zealand, to look at a couple of things. So first of all, who are all the fungi inside Douglas fir seeds? And then do these fungal communities vary by seed provenance or seed region of origin? And this research project inspired my interest in seed microbes and the many mysteries that are still present about them, which I'm excited to talk to you about today. So now that we've looked at the importance of seeds and their microbes, I wanted to talk a little bit about what seed microbial communities look like, based on what I've found in my college research and what I've read about in the past few years. But before we dive into what these communities look like, I briefly want to talk about how we actually study seed microbes. So there are two major approaches. The first is the culture-based approach or culture-dependent approach. And in this method, what we do is you collect seeds from your plant of interest and you plate these seeds in petri dishes on media, which is a combination of nutrients that microbes can grow on. And you leave those seeds in the petri dishes for a while. And then as microbes grow out of them, you isolate those microbes onto separate petri dishes. And then you can use these microbial isolates for DNA sequencing to find out who the microbes are, or for experiments to look at how they interact with each other or what traits they have. And the other major method is the culture independent method. And in this, what we do is we take seeds from the plant of interest and we extract DNA directly from the seeds, microbial DNA. And then we take that microbial DNA and we sequence it in a machine like this one, in what's called next generation sequencing, which sequences the DNA from a bunch of samples all at once. And then with that sequence data, we can do more complicated analyses to assess how microbial communities differ from each other between individual samples or groups of samples based on what the question is. And I used both of these methods in my undergrad research on Douglas fir seed fungi. So the first thing that I learned in my work with seed fungi of Doug fir and of poplar was that seed microbial communities have a few members. So this bar graph shows the number of microbes that my lab mate Sabrina Heitman, our collaborators, and I isolated from individual poplar seeds and across multiple sites in Oregon and Idaho. And as you can see here, we isolated 0, 1, or 2 microbes per individual seed. And the vast majority of seeds had 0 microbes, with a few having 1 microbe, and then very, very few having 2 microbes. And this was also seen in the Newcomb et al. Study, where they looked at microbes isolated out in culture from individual seeds across 98 plant species in these families here. And they saw the same pattern, which is that most seeds have 0 microbes in them, a few have 1 microbe in them, and even fewer have 2 microbes in them. And this has been seen in other culture-based studies as well, which contrasts with culture-based studies on leaf or root microbes, where you may get 15 or more species per individual leaf or root. However, it's important to note here that the microbes found in these studies are those that could grow out on the media or nutrients that were provided. And so there might be other microbes present that just aren't able to grow. And that's where culture independent or sequence-based methods come in. And in my work on Doug fir seed fungi, I got to sequence fungal DNA directly from individual seeds, and I found a similar pattern, that there are few members per seed. So this is a bar plot of all the fungi that I identified by sequencing, and their abundances on the y-axis here. And in the top plot, We have all of the really common or dominant fungal species, which are color-coded here. And then in the bottom bar plot, we have rare fungal species. And what we found was that individual seeds contained 1 to 26 fungal species, which is many more fungi than we've seen in culture-based research, including the culturing work I did with these Doug fir seeds. However, we also found that some seeds, as you can see here, contain 1 to 2 really common or dominant fungi, while other seeds host many rare fungi. And while we found more fungi per seed than in culture-based studies, the general pattern is that seeds still contain fewer members than an individual leaf or root might. And this has been seen in other studies as well. So this is a bar plot from Reski et al., where they were looking at the seed bacteria and seed fungi of radish. And as you can see here, across the seeds that they sampled through multiple sampling years, they found that there's 1 or 2 bacteria that are really, really dominant across seeds, and then 1 or 2 fungi that are really, really common or dominant across seeds. And this makes sense because seeds are very, very small habitats, right? And they have that endosperm, that package of nutrients to nourish the embryo. But that's a very small packet of nutrients. And seeds are also very dry to keep them viable while they're sitting in the soil. So it's a very dry, very small environment. So we wouldn't expect to see a lot of microbes present. Additionally, the next thing is that there are several fungi and bacteria found commonly across seeds in many plant species. So the 2 most common fungi that we see are Cladosporium, which is this green mold here. This is a microscopic structure of Cladosporium, and it's a mold that's found also in leaves, where it parasitizes rust pathogens. So it's a beneficial microbe. And then the other fungus is Alternaria, which is this darker green mold here, which has different microscopic structures. And it's commonly a plant pathogen, so more of a negative microbe. And then in terms of bacteria, the 2 that are most common are Pentoia, which is this one, which can also help in disease resistance, and Pseudomonas, which can be pathogenic, but can also be a neutral or positive microbe. Yeah, so these are some of the common players that we see. And then the other main thing that I found in my research on Doug fir fungi, seed fungi, was that these communities they vary a lot in correlation with a variety of factors. Like I mentioned earlier, I was interested in how seed provenance was associated with fungal community composition. And so to analyze that, we produced this plot, which is called an ordination plot. And to make this, you calculate the differences between microbial communities in your samples, and then you plot those differences in a 2D plot like this one here. And how close or how far away the points are from each other shows how similar or different the microbial communities are. And we found that fungal community composition is associated with seed provenance. So as you can see here, the seed communities from Washington and Northern Oregon are pretty similar to each other, whereas the communities from Southern Oregon, California, and New Zealand are pretty different. And this has been seen in other papers, in other projects as well. For example, Kleidka et al. Found that terroir, which is the combination of local environment and land use practices in agriculture, was associated with variation in seed, fungal, and bacterial communities in bean. And seed microbial communities can also vary across plant populations and even individual plants. So in this study, Fort et al. Were looking at the fungal communities in acorns across several mother trees and plant populations in France. And this is another ordination plot where they've color coordinated the communities by mother tree and then shape coded them by tree population. And they found that fungal communities varied in correlation with both plant population, which you can see by the shapes being clustered together, and mother tree, which you can see by the clustering of the different colors. And this is interesting because it shows how the mother plant can control the microbial communities of their offspring, either by deterring pathogenic microbes or recruiting beneficial ones. And it's also of interest because there's interest in breeding plants to recruit specific microbes for improved health. So seeing this association is useful. And so based on what I learned in my undergraduate research, I came to this question: how do microbes get into seeds in the first place, and what factors are important? And this is the general topic that I'm studying in my PhD research. And it's a really an area that is still developing in plant microbe research. So it's a very exciting place to be in science right now. So broadly, microbes enter seeds via three main routes. The first is vertical transmission, and this is where microbes move from other plant tissues like a leaf or a root up into the ovule and then into the seeds. And this is where we get that multi-generational microbial community. The next route is floral transmission, and this is where microbes land on a flower, or specifically the stigma of a female flower, which is where pollen lands and then germinates to fertilize the ovules. And microbes land on this stigma and then travel down into the ovule and then into the seeds. And then the third route is horizontal transmission, where microbes land on the surface of the ovule or the resulting fruit and travel through the fruit tissue into the seeds. And horizontal transmission can also be colonization by microbes after the seed is dispersed and it's sitting in the soil. And I'm particularly interested in the floral transmission process. All three of these routes can have some quite complicated mechanisms potentially going on. But for my research, this is what I'm studying. And there are a variety of factors at play in this transmission process, and they include the environment, such as climate factors. They include plant traits, microbial traits, and then also dispersal by the environment or by animals. All of these affect transmission into seeds. And I want to note that these factors all have different effects at various spatial scales. Spatial and time scales are a real major focus of ecology because processes occur differently at different scales. And as I talked about before, seed microbial communities can vary across sampling sites, plant populations, and individual plants, which typically occur at these really large scales of the macro scale, which is many kilometers or across a landscape, to the mesoscale, which is meters to several kilometers. However, my PhD advisor, Dr. Johan Leveau, and others have noted that microbes behave at the very, very small scales, or the micro scale, which can be nanometers to centimeters, or in the different parts of a seed, or across different tissues of an individual plant. Kreger et al. Also demonstrated that microbial communities vary across different organs or parts of a whole individual plant. So it's important to consider all of these scales, especially because some factors may be more important at a large scale versus a small scale, and then different processes may be at play at each scale, such as the mode of dispersal at the very, very small scale versus the larger scales. And so to dive into these factors, I first want to talk about abiotic environmental factors. And some of the main ones that impact microbial communities are temperature, water availability, and ultraviolet light exposure or UV exposure. So this is a figure from Zimmerman and Vitusik. And in this study, they were looking at fungi growing on leaves across rain and temperature gradients around the Mauna Loa volcano in Hawaii. And this is another ordination plot looking at the fungal communities, along with a correlation to the rainfall and elevation gradients across this landscape, which are shown with these arrows here. And they found that community composition was associated with water availability or rainfall and temperature and elevation. And at smaller scales, temperature, water availability, and UV exposure may also impact microbial survival and growth, such as on the surface of a flower. So this brings up a really important question. A lot of these environmental factors are studied at large scales of many kilometers, and they've been looked at primarily for leaves and sometimes for roots. But as I mentioned before, my Dr. Laveau and others have noted that microbes behave at the very small scale. So how do these factors impact seed microbe survival and growth at very small scales? One scale of interest is different locations on a single plant, such as different heights in a tree's canopy. For example, Harrison et al. Were looking at how canopy position impacted the fungal communities of leaves in coastal redwoods at these 3 different heights. And they found that the communities do vary with canopy position and could be associated with differences in light, temperature, water, and UV exposure at different points in the canopy. This hasn't really been looked at for seeds before, so I think it'd be really cool to do a study where you sample seeds from different positions on a tree and also collect environmental data from these different positions and see if there's a correlation between the microbial communities and those factors. Going to an even smaller scale, how might seed microbes be affected by different environmental conditions on a flower before traveling into a seed? So in this study, Hayes et al. Were looking at the bacterial communities on different parts of sunflower petals for 2 different sunflower species. And they found that bacterial communities did differ a little bit in association with where they were on the petal. And they also found that there were different levels of UV light absorption along the length of these petals, particularly for this species, and that bacteria had different levels of survival ability against UV exposure, and that was correlated with where they had been found on the flower petal. So it's important to think about how these abiotic factors might be at play at these very, very small scales. On top of environmental factors, we also have various plant traits. And the ones I want to emphasize are plant defenses, nutrient release, and surface topography, which can also impact microbial survival, growth, and transmission. So plant defenses are really important during seed production. Seeds are one of the most defended plant tissues because they are agents of reproduction, and they can be defended by physical barriers in addition to chemical compounds. And there are these chemical defenses present in flowers. Additionally, there are a variety of compounds on the surface of the stigma where microbes might be transmitted to seeds. And these include sugars, which are shown here in blue, fats here in orange, and proteins in green. And different microbes are going to be able to use different nutrients and be able to respond to different plant defense compounds. And so understanding how these different microbes are able to use nutrients and evade defense mechanisms is really important. Additionally, plant surfaces are very variable. They have lots of ridges and valleys, and it's been shown that this variability in plant surfaces impacts microbial communities. This has been shown in leaves by Doan et al. And it's been shown in flower parts too. So this is a microscope slide from Spinelli et al., where they were looking at bacteria on the stigmas of apple and pear flowers. And as you can see, there are all of these bumps on the stigmas, which create mountains and valleys on the stigmas effectively. And you can see the bacteria glowing here in green and red, moving around in the valleys between these bumps. So it suggests that the surface topography of flowers is another important factor in how microbes are colonizing flowers, surviving, growing, and then eventually ending up in seeds. But these microscale plant traits haven't really been looked at in relation to seeds yet. And so, as I mentioned before, stigmas are really very bumpy micro landscapes, and that microbes are interacting with this micro landscape. Additionally, Iyer et al. Found that that microbial communities can differ between the tissues in a seed. Fort et al. Also showed this, so this is another microscope slide of those acorn fungi, and you can see the fungi growing in the different tissues of the acorn in these blue and green patches here. But the stigma is a very variable environment, and then the different parts of the seed are very different as well. And so it's important to think about how this variability across the very small landscape of the flower and the seed might select for certain microbes over others. And then on the flip side of that, the other question to think about is what microbial traits are involved in transmission to seeds. And to do this, I'm looking to do an experiment in the future with Acidivorax citrulli, which is a bacterial pathogen of watermelon. And it's been shown that this pathogen can infect seeds through flowers of watermelon. And so it serves to be a really good positive control for looking at what traits are necessary for a microbe to move from a flower into a seed. And to do this experiment, what I'm going to do is create different varieties of Acidivorax citrulli that have some traits turned on, some traits turned off. And I'm going to inoculate a mix of these bacterial varieties onto watermelon flowers. And then after the fruits develop, I'm going to harvest the seeds, directly sequence the microbial DNA from them, and figure out which varieties actually ended up in the seeds versus not, and what traits did those varieties have, and what function those traits might be serving in transmission. And microbes aren't interacting one-to-one with plants. They're also interacting with each other during this transmission process. And a lot of these interactions have been described as competitive. So for example, in Cui et al., they were looking at how inoculating apple flowers, like these shown here, with the fire blight pathogen, Erwinia amylovera, impacted the bacterial communities on the floral tissues. And so this is a bar plot that they produced, similar to the ones I've shown before, where they color code the different bacterial species or bacterial families here, and their abundances are on the y-axis. And they show that after inoculation with just water, bacterial communities become dominated with Pseudomonas or some Enterobacter, But when the flowers are inoculated with that pathogen, Erwinia amylavora, the pathogen becomes the dominant member of those communities. And so there's some sort of competition going on where Erwinia amylavora is beating the other microbes in that community. And this has also been seen with fungi. So in Raghavendra et al., they were looking at, they were inoculating flowers of Centaurea stebe with combinations of fungi and then re-isolating those fungi out of seeds. And they found that only one fungus from the combinations was re-isolated out of seeds. And it tended to be the same fungus, which shows that there's some competition going on as well, and some fungi are better at competing than others. However, microbes can also coexist inside seeds. So in this study, Torres Cortes et al. Were looking at how microbial communities in seeds changed when they were inoculated with a fungus and with a bacterium. And they found that compared to the uninoculated seeds, the communities aren't super different from each other, as you can see looking at the colors of the bars here. And their explanation for this was that the other microbes in the community were using different resources than these two microbes that they inoculated seeds with. And so the question I have based on all of this is, how do these microbes actually sense and interact with each other during transmission into seeds? We've seen that sometimes they compete with each other and other times they coexist, but the how is not really known. So it is known that fungi can sense hormones, sugars, and proteins, and even gases in their environment. And bacteria can also sense those compounds and then certain compounds given off by each other for communication. So how are these microbes actually sensing each other? What dictates them competing versus sharing these resources? Do they use the same nutrients or not? And does this have any relationship at all with moving into the seeds? Finally, the last major process I want to talk about is dispersal, and dispersal is the movement of organisms between different habitats. And seed microbes can disperse via a variety of processes or modes. And at the really small scales, they can disperse via their growth, water, and wind. So this is another microscope picture from Spinelli et al., where they were looking at those apple and flower stigmas. And you can see the bacteria growing down the surface of the bacteria here, as shown glowing in green. So growth via production of cells or moving of growth of new fungal filaments or hyphae can be a form of dispersal at these very, very small scales. Microbes can also be moved by water, such as these fungal spores being moved by water droplets, and they can be moved by wind across very small distances to very large distances. So this is a map from Pei et al., where they were looking at the movement of fungal spores across a landscape. And you can see there are millions of spores, and they're able to move across this landscape, some at short distances and others at really long distances. And seed microbes can also be dispersed by animals, including birds, and then pollinators such as bees. And one major really cool study that I want to tell you about is Prado et al., where they were looking at the movement of seed microbes by honeybees and by mason bees in canola, which is this yellow flower here. And so they had this greenhouse set up where some of the greenhouse rooms had bees and others did not. And they hand pollinated some flowers and let the bees pollinate other flowers, and then looked at the seed bacterial communities. And they found that the bacterial communities included bacteria that are often associated with bee gut, the bee gut community, and that these bacterial communities differed quite a bit between the bee-pollinated seeds, which are shown here in yellow in both panels, versus the hand-pollinated seeds shown here in green. And this was the first time that is demonstrated that pollinators can move seed microbes around. It's been shown for a long time that pollinators move flower microbes around. So it was really cool to see this connection through the flower to the seed. However, that experiment was conducted in a greenhouse. And so the question that I have is, how do pollinators impact seed microbe dispersal across large-scale wild landscapes? So pollinators have different ranges based on their wingspan. They also have different preferred plants that they like to get pollen and nectar from. And so how are different pollinators moving microbes around within a particular patch of flowers? And are they able to move microbes around between patches of flowers that are close to each other and then patches of flowers that are very far away from each other? And this is another question that's been explored a little bit with flower microbes, but not with seeds yet. And I hope I can do a study on this question, because it's a really cool question from a microbe perspective, but also a pollinator service perspective. So finally, I want to touch on what microbes are doing once they're inside seeds. And this is outside of what I'm doing in my PhD, but it is another area that has a lot of complexity and a lot of questions associated with it. So I wanted to talk about it briefly. The first major question is, how do animals change microbial communities during seed dispersal? So after maturing, seeds are dispersed to sit in the soil, and a lot of them are dispersed by animals such as birds or insects or squirrels or even large animals like bears. And Correa et al. Showed that birds can disperse fungal spores and seeds at the same time. And Lash et al. Also found that ants who bring seeds into their nests before depositing them out in the soil, they also change the microbial communities in and on seeds. And so this is just very preliminary evidence that animal dispersers are changing these communities, but it's not really understood yet how this is actually happening, and if the changes that are made during movement by insects or sitting inside a bird's gut, how that impacts the seed community and the resultant seedling community. And then the other major question is, how do seed microbes interact with soil microbes during dormancy and germination in the soil? So seed germination in particular is a really chemically active time. The seed is taking on water and it's releasing sugars and other compounds into the soil, and microbes respond to the release of these compounds by growing really rapidly in the soil. But you also have a couple of microbes inside the seed to begin with, and it's not really known how these microbes are actually interacting with each other. And it's important to think about this question because eventually those seed microbes and soil microbes are going to be seedling microbes, whether in the leaves or in the roots. And it's been shown by Doherty and by Johnston-Monge et al. That seedling microbes are sourced both from the seed and from the soil. And the majority of microbes inside leaves and inside the root, not around it, come from the seed itself. So the how of this process is still up for study. And this is another really important question because ultimately these microbes are going to be affecting the seedling and its health and function. So to conclude, I want to paraphrase the science journalist Ed Yong and say that seeds contain multitudes. They are an ecosystem unto themselves. So individual seeds are home to a few fungi and bacteria and maybe some other microbes. And these microbes can be transmitted into seedlings, impacting early plant health. And while the process of transmission to seeds is generally known, the details of that process still need to be explored. And studying how that transmission occurs will help us better understand microbial communities and maybe even design them for improved plant health. And with that, I wanted to give some acknowledgements. So thank you to my advisor, Dr. Johan Laveau, and my lab mates in Lab Laveau for all of their feedback and support. I also want to thank Dr. Posey Busby and the Busby and Laboldis labs at Oregon State University, which is where I got my start in plant microbe research. And thank you to the Sonoma County Mycological Association and the National Science Foundation for funding my education and my current research project. So thank you. Do you have any questions?
We did have one question. Maybe that was some of your continuing questions on the unknown of the process of transmission. But how did we figure out that microbes are transmitted into seeds by floral, by those three different mechanisms? How did you figure that out?
A lot of it was done through looking at seed pathogens. So that reference I gave is a whole book about seed pathogens and how to fight against them. And so basically there were just experiments on, we see these seed-borne pathogens, how are they getting into seeds in the first place? And like, for example, in my study system, Acidovorax citrulli and watermelon, they found that the bacterium was getting into seeds via flowers by inoculating flowers with bacteria and then checking to see if the seeds had that bacterium in them. So it's studies like that where you could inoculate a leaf with a microbe of interest and then check the seeds for that same microbe, or just inoculate the outside of a fruit and then look at the seed afterward and see if that microbe is there. And again, like, a lot of that has been done with pathogens because for agricultural reasons or disease, like plant pathology reasons, we're really interested in that to prevent the spread of seed-borne diseases. But now I think we're starting to get at why, like, looking at how other microbes might be doing the same thing and how that might be a beneficial thing. Yeah, I hope that answered your question.
No, it's good. And we have a couple questions. Wow, thank you. Does this have anything to do with seeds' ability to adapt to climate change?
A little bit, yeah. So it's been shown that various microbes can improve plant responses to stressors like drought, which is a really big problem associated with climate change. I live in California, where we've had a drought for years. And so dealing with water management in agriculture is a really big issue. I found in my undergraduate thesis, and it wasn't like a super significant result, it was just a trend, that when I inoculated seedlings with fungi, the ones that were inoculated with fungi survived drought conditions slightly better than the ones that were not inoculated. And so we can, so the ideal out of plant microbiome research as a whole, and this includes seed microbes, is, can we find beneficial microbes consistently across plants that improve drought tolerance or other stress tolerances, like tolerance of disease, which is another thing that's going to become more prevalent with climate change, disease epidemics. Can we find microbes that improve plant tolerances to those kinds of stressors so that they can grow better in the face of climate change? Yeah.
We have another one here. Central Oregon, is beer country. Mm-hmm. Could it be that engineering the seeds for hops, barley, etc., could have important economic impacts by changing the beer flavor or acidity, etc.?
That's a really good question. thinking about barley in particular, I heard about a study the other day where I think I'm trying to get this right, where plants, I think it was like barley, or no, it was wheat. It was for bread. Varieties of wheat inoculated with different microbes had distinct flavor, either because they were able to grow differently or accumulate different nutrients because of the microbes. And so I think you could have a similar result with barley. I'm not so sure about hops, just because it's a different plant form. But for barley, for sure, it could change. It could change the flavor profile of the barley itself.
Well, thank you, Jillian. You've given us a lot to think about and digest, and I'm so happy you were able to join us. Mm-hmm. So have a great rest of your evening, everyone, and enjoy.
Thank you. Yeah, and if you have other questions, feel free to email me. I'm happy to take them.
Wonderful. Thank you for being a resource.
Yeah, of course.